to PK Tech Lab. Today, we'll be talking about the compound light microscope. If you would recall, in our previous video, we made mention of the fact that the microscope is one of the basic instruments one would find in a biology lab, especially a plant anatomy and physiology lab. So today, we'll go a little more into details about the microscope. At the end of this video, we should be able to identify the different parts of a compound microscope, and we should be able to know the function of each of these parts. So first of all, why is it called a compound microscope? It is called a compound microscope simply because it uses multiple lenses. Going forward, we will understand this use of multiple lenses into more details. And why are we saying it is a light microscope? Because it uses light as its source of illumination. Okay. So how powerful is the compound light microscope? I will say that it is powerful enough to make invisible objects visible to the human eye. It is able to enlarge minute micromolecules enough for us to see with our naked eyes. That is how powerful the compound microscope is. In fact, without the microscope, a whole world would have been unknown to us. It is because of this compound microscope that we have been able to discover the world of microorganisms and even know that they exist. Without this microscope, we wouldn't have known that minute life forms existed. Maybe we would have felt they existed, but we wouldn't have had any evidence for them. So that's how powerful the microscope is. So now, what are the parts of the compound light microscope? We have the ocular lens, which is the eyepiece. That is what we see at the top here. And then we have the revolving nosepiece. That's the dark part we see here. We have the objective lenses. They are actually 4 in number. We'll see them more into details later. Then we have the stage with clips. So the dark side we see here is the stage, and then the silver-like metal we see here is the stage clip. Then we have the condenser and the iris diaphragm. They are down here. They are not very visible in this picture, but we'll see them more into detail as we go along. Then we have the illuminator, the light source. It's labeled here. And then we have the coarse and fine adjustment knobs, which we see here. Then we have the brightness adjustment knob or switch, which is visible here. And then we have the on and off switch. Good. So what are the functions of all of these parts that we've stated here? The ocular lens is where we look through. We look through to observe the specimen on the stage. So we place our slide on the stage. We look through the ocular lens and observe the specimen on the stage. The revolving nosepiece is used to change the objective lens that we are using. So when we rotate this dark one, it could change to times 10 times 40 times 100. And that is the work of the revolving nose piece. The objective lenses are of different magnifications. The higher the magnification, the bigger we see the specimen on our microscope. So we'll talk more about that as we go along. Then we have the stage. The stage is where we place the slide. The stage holds the slide, and the clips hold the slide firmly in position. The condenser and the iris diaphragm. The diaphragm is used to control the amount of light that passes through the condenser to the specimen on the stage. The condenser is the part that focuses the light from the illuminator onto the specimen on the stage. So the condenser focuses the light, and then the diaphragm controls how much light is focused, controls how much light is focused. Okay, so we can understand the different functions of condenser and iris diaphragm. We'll see them a little more clearer as we go along. The control knobs, the stage control knobs, are used to control the stage here. So the knobs are two. There's one above and there's one beneath. The one above is used to move the stage forward and backwards. The one below is used to move the stage sideways. So they are used to control the movement of the stage. The control knobs, coarse and fine adjustment knobs, also are used to control the stage, but they move the stage either up or down. So that's the difference between the coarse adjustments, fine adjustment knob, and then the stage control knobs. The on and off switch is obvious. It's used to turn the microscope either on or off. So these are the parts of the microscope. Let me not forget to mention the arm. This arm helps us carry the microscope from one place to another. So we hold the arm with one hand, and with the other hand we hold the base of the microscope. So this is the base of the microscope for transporting it from one end to the other. Okay, so these are the parts of the microscope that we need to know to be able to operate the microscope efficiently. So now let's watch this brief video that gives us a more closer look at each of the parts that we have mentioned. So let's watch as I explain along. Okay, so at this point we could see the fine adjustment knob clearly, and then the coarse adjustment knob. Now what's the difference in their function? This is coarse adjustment, this is fine adjustment. The coarse adjustment knob moves the specimen, is used to focus the specimen by moving the stage up and down. It puts the specimen into focus. After putting the specimen into the initial focus, the fine adjustment knob is used to make the image sharper or clearer. So that's the function of these parts of the microscope. So let's go along and view the other parts. What we see here are the objective lenses. The objective lenses are in view here. If you observe closely, you will see that the objective lenses are color coded and they have their magnifications etched on them. So what we have in view right now is the ×4 objective lens. It is red color coded. This is actually a Leica microscope, so you can see that this is the good ×10 objective lens. Sorry, my video has moved back. Okay, let's continue. Right, so ×4 red color, then we have the ×10. Good. So we have the ×10 showing the yellow color coded. I hope we've seen that. Then we have ×40 blue color coded, and the ×100 we saw previously, that was cream. The higher the magnification, the longer the objective lens and the bigger it is. Okay, now what we have in view here is the iris diaphragm and the condenser. The iris diaphragm, we move it to the left or to the right to adjust the amount of light that is allowed to pass through the condenser. The condenser, there is a system of lens inside here that focuses the light that passes through onto the specimen on the stage, which is further focused onto the ocular lens and then finally focused onto our retina for us to see the image. So what we see here, the condenser and the iris diaphragm. Then we have the illuminator. We have the on and off switch, 0 for off, 1 for on. This is the base of the microscope. And then we have the knob for adjusting the brightness of light that we are allowed through. So these are the objective lenses, the ocular lenses that we look through. These are the ocular lenses. If you observe carefully, you will see that the ocular lens has a magnification of ×10, and this is the arm, the cord that we connect to a light source. So these are the parts of the microscope, and so I hope that with all we have learned, when you meet any light microscope, you will be able to identify the parts and the functions that they play. Thanks for watching.